Hello everyone, I'm Dr. Juan Du from Shanghai Changzhen Hospital, China. I'm honored to present updated results with phase 1 open-label single-arm study of dual-targeting BCMA and CD19 FASTCAR-T GC012F as a first-line therapy for transplant-eligible newly diagnosed high-risk multiple myeloma. As you know, the current approved therapeutic options have benefited myeloma patients, but prognosis among high-risk myeloma patients remain poor, with a median survival is only about 3 years, even in transplant-eligible patients. So we really need a novel, effective first-line treatment to improve clinical outcome in this subgroup. GC012F is a second-generation BCMA CD19 dual-targeted CAR-T therapy targeting both antigen-centric elimination of myeloma plasma cells and CD19 positive progenitor cells to drive faster, deep, and durable response in multiple myeloma patients. GC012F is manufactured on the novel FASTCAR platform, enabling CAR-T manufacturing time to next day. The platform was designed to overcome major challenges of conventional CAR-T therapy, such as the length wait time, T-cell exhausted, one of the outstanding advantage of the platform. Another advantage is enhanced CAR-T cell quality and a high concentration of the young phenotype T cells. This is a study design. This is a single-arm open-label phase 1 IIT study. Here, the eligible criteria. The newly diagnosed patients enrolled at least one of the high-risk factors. From the August 2021, patients continue to be assessed for response. The date cutoff April 23, 2024. So the right of the slide, you can see patients received 2 cycles of VRD induction therapy. We have treated 22 patients with a different dose level from 1 to 10 times 5th power to 3 times 10 to 5th power. The primary of the study was the safety, and the second endpoint including ORR, best response, MRD assessment, and PK and PD. This slide shows the patient's characteristics at the diagnosis. You can see the patients in this study has a very aggressive disease, with 91% of the patients in revised stage 2 or 3, and 91% percentage of patients with a high risk profile according to the MSMART 3.0 criteria. 52% of patients having a high-risk cytogenetics, and 55% of the patients having an extramedullary disease. They actually has a very high rate of the high-risk features. You can imagine patients might be have the inferior outcome with a routine frontline therapy. So here we report the safety data of this study. As you can see, the most common grade 3 or 4 adverse was a hematological event, which was expected. Most common hematological adverse event was leukopenia, lymphopenia, and neutropenia. Anemia and thrombocytopenia also quite common, but they recovered very rapidly. Non-hematological adverse event was mainly grade 1 or 2, the most common being LDH increased and hypercalcemia. Looking at the detail of the CRS, only 6 patients, 27% experienced low-grade CRS. 5 were grade 1 and 1 grade 2. The median on-site was 7 days for patients with a median duration of 1 day. We have no patients grade 3 or high CRS or ICANS, no neutral toxicity in this study. The one thing I'd like to point out is CAR-T in newly diagnosed patients, CRS could be resolved within 4 days. So this is a very safety profile. Looking at treatment response, we saw a very high response rate, with all patients' response to treat at 100% overall response rate, with a SCR MRD negativity of the 95%. The median duration of the follow-up more than 2 years, the median duration response DOR, and the median progression-free survival was not reached at a day cutoff. This slide shows you the MRD results. We also saw very high-risk MRD negativity, 100% evaluable patients at 1 month, 95% at 6 months, and 12 months. This is measured by next-generation flow cytometer with a threshold of the 10 to -6 in all dose levels. Here's the pharmacokinetics. In terms of the pharmacokinetics, GC012F expanded very well in all patients in all dose levels. The peak happened about 10 days. The median peak copy number nearly 60,000 copies per microgram genomic DNA, and we observed a median persistent about 29 days. So in conclusion, GC012F saw a very favorable safety profile in newly diagnosed patients, and was successfully manufactured for 100% patients using a novel FASTCAR-T platform. GC012F, we observed a very high quality safety, almost low-grade CRS, and there were no ICANS or any neutral toxicity observed. Especially, this drug product demonstrated GC012F achieved a high response rate, ORR of 100%, VGPR, and 95% SCR in high-risk population. It also produced very high MRD negativity rates in evaluable patients at 100% by EuroFlow assessment at a sensitivity of 10 to -6. It is able to point out we saw a fast and deep response with a median DOR not reached. So we think GC012F, BCMA and CD19 dual targeted CAR-T show a very encouraging anti-tumor activity in transplant-eligible, high-risk newly diagnosed multiple myeloma patients. Thank you.